I am the chair for the session this morning. My name is Brett McGuire. My name is on the slide. I'm giving a warm-up talk here. I also have the privilege, since we didn't plan ahead, to welcome you all to the conference, the 10th annual Astrobiology Graduate Conference. It's a really fantastic opportunity. We're going to get to hear a lot of talks crammed into 2 days, and then FAME Lab or the field trip tomorrow. Be a blast. One of the reasons we have these warm-up talks historically is because this is a really, really broad audience, right? And so if you hear me joking around here, I'm giving a warm-up talk on a subject I'm not even familiar with, half of the talk. So the purpose of these, and there'll be one either in the morning or the afternoon, and one of them is split up even between two sessions because they're so diverse, is just to give you a flavor of the kinds of things that are going to be talked about in that session. So for this one, solar system bodies here. It's the second session this morning. I don't know anything about this. So what I did is I just went to Wikipedia and found all the things that I found cool about Mars and Europa and said, oh, maybe people will be interested in that. So that's what we're going to do. I don't know what the other warm-up talks are going to be like, but that's the general idea to give you a flavor. But this is also the warm-up talk for the entire conference. So I thought I'd give you just a brief preview of what the people that organize this conference are like. So you can get an idea of how this is run. So as you can imagine, we've sent a lot of emails back and forth over the last few months, and they have degraded in quality and robustness, I guess. So here's a sample of the one-line subjects, one-word subjects that have been sent. That's just in the last 2 months, which is pretty fantastic. And we also recently devolved into expressing our feelings through memes. And this is just a small selection of those. We also have been all assigned characters from Firefly, which fit our personalities and roles in this conference, apparently. And just one last thing to introduce you, he'll be talking a little bit later, to our fearless leader, Bradley Burker. Here he is at RFG on the balcony that he locked himself on. So if you have questions, go ask him, as long as you can find him. All right, so there aren't any balconies around here, right, Brandon? Fantastic. All right, so let's see, warm-up talk since we started a little bit late. And feel free to interrupt me at any time if you have questions. It doesn't bother me whatsoever. So the first half of this is, we'll do this. Never mind. I wrote this at RFG yesterday. So, I'm a Caltech graduate student, and at Caltech we just started a summer seminar series with professors giving talks on their science, which is something that doesn't happen very often, actually. You don't get to hear your professors give talks. And we've had 2 so far, and they've been really great, and they both brought up this idea of great questions in science that they think are going to be solved over the next 30 to 50 years. These are the big things that have to be addressed. And one of them, a guy named Harry Gray, he's a riot, he talks about splitting water here. Does this work? Oh, this is not very bright, Bradley. All right. Splitting water to make energy from solar light. All right? This is the next great advancement in human civilization and energy, according to Harry Gray. Because without this, we're just going to run out of fuel to power our lives, and that's not very good for us. Hey, that's much better. All right. Then we had another talk, a guy named Dennis Doherty, and he said, other great question is the human mind. So understanding how we work, right, from a molecular level. So he works on signaling pathways in the brain and understanding neurotransmitters. All right? And he says, this is the most complex piece of machinery on the planet, is the human brain. So 2 great questions are, how do we work, and how are we going to continue to survive? And I think this may be presumptuous from a graduate student, but a third great question is, when are we going to find alien life? Are we alone in the universe? All right? And this has general implications for the conference, right? This is astrobiology. But it encompasses a whole range of topics, and we're going to talk about all of them today, right? It's not so specific, say, as splitting water, but how did life arise on our planet? How might life arise on other planets? How might we find it? And how do we understand the evolution of that life? Now, let's see if we get to the slide I thought was coming next. Here it is. All right, so the first session of the day is focusing on astrochemistry. This is what I do, so these will be a little bit better than the coming slides. So I define astrochemistry to my friends when they ask is the study of molecules in space, where they are, how they got there, and what they're doing. All right? So looking out into space and cataloging in a range of evolutionary stages what molecules are present and how do they lead to the evolution into subsequent steps. All right? So if you start off all the way back with the Big Bang, you didn't make molecules. You made hydrogen, helium, and a smattering of lithium and beryllium, and that was it. And that was it for a very long time until the universe expanded and cooled off, and then that hydrogen and helium condensed down to form the first stars. Right? And then those nuclear furnaces, they fused the hydrogen into helium and into heavier elements until the stars themselves died out, went boom, and distributed heavier and heavier elements into the cosmos. And when those boom clouds, right, the remnants of these supernovae, expanded enough and cooled off enough again, that hydrogen could start to condense itself, form the first molecule, H2, right, and then start a chain of reactions from there. And then you can trace that evolution from those early molecular clouds through the formation of subsequent stars, which process heavier and heavier elements, right, and make more and more complex molecules as they condense out and eventually form solar systems and us. And the study of the chemistry tracing through all of these molecules is what astrochemistry is. So astrochemistry as a field, actually, you go all the way back to 1937, which doesn't seem very far because it's a very young field. The first molecule wasn't detected in space until 1937. It's the CH molecule. And then between 1940 and 1963, we only found 3 more, all right? CN, CH+, and OH. And it wasn't until the 60s, and this is when radio telescopes and infrared telescopes really took off in their evolution in technology, sensitivity and resolution, that allowed us to look further, deeper into these clouds. And we found 59 more molecules in the span of, what's this here, 20 some odd years. This is the real birthplace of astrochemistry. This is where it emerged as a discipline and people started using this actual word. And since the 80s, we've detected more than 110 more molecules. So now there's over 180 individual molecules. This is not counting isotopes, right? This is just regular molecular species detected in interstellar space, just floating out there in clouds. These are very complex molecules, more than 10 atoms in some cases, and in special cases with buckyballs, C60 and C70, right? These are dozens of atoms floating out in space that have come together in the vacuum of space, and they're doing chemistry. I think that's really cool. So astrochemistry itself can be broken down into 3 subdisciplines, and you'll get a little bit of all 3 of these today, which is really cool. In 4 talks this morning, you're going to get some representative from each of these in one way or the other. Laboratory astrophysics, and see there's Marco, wrong one. There's Marco up there over one of our laser systems in the lab. All right, so if you want to compare your observations that you're taking with the telescope, figure out what molecules you're seeing, you have to actually first study those molecules in the lab. All right? So you have a reference to identify, this transition showing up at this frequency is coming from this molecule. So that's what you do in the lab in the very most basic types of laboratory astrophysics. But you can go further than that. If you use your astronomy and you identify that there are molecules X, Y, and Z in various abundances and temperatures and densities in these sources, right, then you might want to know, well, what can I make from those? Right? So we give that information to astrochemical modelers, and they say, well, that's fantastic. You told me what we have, but how do they react? So the laboratory astrophysicists go back, and they put these things together in a reaction chamber, and they give you rate constants and energy barriers. And they say, well, they react to form this molecule this fast with this kind of reaction energy. The modelers put all these into complex chemical codes, so hundreds of molecules and thousands of reactions. It's a giant network, vast computer resources, right? And they just step it through in time. And they say, well, you have these molecules. These are going to make a high abundance of this molecule. You should go look for that. And the astronomers say, well, we don't know where that is. So the laboratory astrophysicists say, well, we do. We can find that. And the cycle just keeps repeating itself, and they feed back into one another. And eventually you start to form this large picture of complex chemistry in a source. So why do we, I hope this is the slide next. Yeah, why do we care? Well, this is a picture of a protein. something you might see in your head. In your food last night, it's just a cartoon sketch. But if we zoom in here on one of these little pieces, right, these proteins are just made up of sequences of amino acids, as I'm sure most people know, right? And they have different structures. So there's glycine and alanine and glutamic acid, all in sequence here. And it's the ordering of these sequences that builds up the function of the different proteins into our bodies. We'll come back to why I picked these three out here in a second. So how do you make these complex molecules that make up life? Well, one of the earliest thoughts back in the 50s, as I'm sure you're all familiar, is the Miller-Urey synthesis, right? Start with basic molecules, carbon dioxide, water, ammonia on an early environment, and you just strike it with lightning over and over and over, and you put energy into the system, and eventually it builds up some complexity. And whether or not this actually works on Earth has gone back and forth over the years as we learn more about what our atmosphere was like. But this is one possibility. Start with the simple molecules on Earth and just hit it with lightning. Another possibility was raised once this happened. September 28th, 1969, in a little town called Murchison in Australia, the Murchison meteorite impacted. And we were lucky because the residents of the town saw this meteorite strike and they immediately went out and started collecting samples. So before these things could get contaminated too much by their environment, they were isolated by the townsfolk and then kept waiting in freezers in some cases for scientists to come up and get pristine samples of them. So we have pristine samples from this Murchison meteorite. So shortly thereafter, somebody just scraped some of this off, dissolved it in some water, and stuck it in a mass spectrometer, GCMS. And this is what they found. Alanine, glycine, glutamic acid. All right, those amino acids that make up that protein that I showed you on the previous slide. Found in a meteorite, a pristine remnant of the early solar system. All right, these are not contaminants. We know from the isotopic ratios this did not arise on Earth. All right, so these amino acids, these building blocks of proteins of us, were found in space long before they were on the Earth. That's really cool to me. All right, so another possibility here is that, well, what if we didn't need the Miller-Urey synthesis to make any, or maybe not all, of our amino acids? What if it happened during delivery processes? Well, there's been work to look at whether or not, well, you start with the simple molecules in the comet, or the meteorite, and then when it impacts the Earth, right, there's intense pressure and heat that's generated at that impact. And that's just the same thing as a lightning strike, for better or less, right? And so that it puts energy into the system, and you synthesize all of your biomolecules all at the moment of impact. And of course, the other possibility is, well, maybe they were formed in the meteorite of the comet beforehand, and they just survived impact. And that actually works, too. People have done these experiments in labs, just slamming amino acids at high pressures and temperatures, and they see that they survive. All right, so there's more possibilities for the seeding of this prebiotic material on Earth than we had originally thought. These are very recent papers, this one in particular, and they've just started some experimental work on this. This is theoretical work here. There's also been some independent confirmation that these things actually exist, right? So I don't know how many of you are familiar with the Stardust mission, but NASA took a satellite and flew it through the tail of a comet. Right? This is just passing through the ice and the gas and the dust, which is coming off the tail of this comet as it passes by the sun and gets burnt off. They collected the stuff that came off, shot it back, we picked it up out of the desert, and we found glycine. What's not there? We found glycine. There it is. In this sample. So this is even more pristine of a remnant from the early solar system than that meteorite that impacted. There's no chance for Earth contamination here. All right? Glycine is out there. So one of the big questions in astrochemistry is how do you get from the simple molecules formed in these molecular clouds after the stars have chewed up those hydrogen and helium elements, spat them out, to the amino acids that we see on Earth? Right? And there's different possibilities here. So there we go. Right? One is you just incorporate the simple molecules onto the planet, as we talked about, either while the planet is forming, it just accretes during the older solar system, or it gets delivered by those comets. Do Miller-Urey synthesis, there you go. Another is that those simple molecules get incorporated into the cometary ice. That comet impacts the planet, and that impact event generates the amino acids. Right. Or let's see, you could go the other way here, right? The simple molecules impact and then undergo Miller-Urey synthesis, some combination of the two. We can go the other route and say that the simple molecules are going to stay in the gas phase the entire time, and they're just going to react, do chemistry in the cloud, form amino acids just floating around in space, and incorporate those into the planets. You can also have them incorporated into the ice of the comet and then do reactions in the solid state, in the ice, the molecular water ice on cometary surfaces, and then impact them. We know that the amino acids survive there. Or you can go even a more circuitous route, take simple molecules, put them in an interstellar ice on the surface of micron-sized dust grains, all right, form complex molecules there, have all of that dust accrete, form a comet, and then impact a planet that way. So we know a few things about these different reaction pathways, and one of them is that this route here, forming complex molecules like glycine in the gas phase, just doesn't work. Right? Laboratory experiments proved that there's just too much of a reaction barrier to make anything more complicated than about methylformate, as long as it's an organic molecule. You can build up long-chain hydrocarbons, just carbon and hydrogen and some nitrogen, that's fine, or buckyballs, but the things that we need for life, they're just not energetically favorable above, say, about 8 or 9 atoms total, with few exceptions. So you have to make these things going through solid-state reaction chemistry at some point, either in ices or once you get to the Earth. And the other thing we know is that from the Stardust mission, there are complex molecules present in these cometary ices. Right? So now, for astrochemists, the question is, where along this path do you actually make these complex molecules, and what's the chemistry necessary to do it? Right? Because once you understand that, then you can start looking at these different sources, right? So molecular clouds or protoplanetary disks forming star systems, and say, which one of these is eventually going to lead to the formation of life? What's the place we should be looking for, based on the chemistry that we're observing, to go look for life in other worlds? All right, so moving on to planetary solar system bodies here. This is the wishy-washy portion of the talk. The reason we're interested in that is this really pretty picture here from our proposal at RFG. I put it in there because it looks nice. This is the Grand Prismatic Spring in Yellowstone, right? This is a really unfriendly environment for life as we really think of it ourselves, but there's tons of microorganisms living in here in this extreme harsh environment. And so this leads us to thinking, well, what if these microorganisms can live in other harsh environments in our solar system? Like this little guy here, if you've been watching Cosmos, the tardigrade, right? This is apparently Neil deGrasse Tyson's favorite little water bear. This thing can survive in Antarctic ice. It can survive in the vacuum of space. It can survive incredible doses of radiation. It just goes dormant, and then when it gets a little bit of heat, a little bit of nutrients, it just picks right back up where it was going after decades. Right? These things can survive anything. So if they can survive anything on Earth, why can't they survive anything or something like them on another planet? There's our solar system. Pluto should be out here. Today, the talks, fortunately for me, only focus on a very small region of our solar system, Mars and Europa, a satellite of Jupiter here. So, as I said, I just went to Wikipedia and looked up things that I found fun about Mars and Europa, so we'll just blast through a few of these here, and then we'll open up the floor to questions. So there's Mars. Mars is about half the size of Earth, if you've never seen the comparison. It also has a slightly off-kilter orbit from Earth, right? So we actually pass closer and farther away from Mars every 2 years or so. The closest approach to Mars, I think, was a couple years ago. But in 2018 here, we're actually only going to be 57 million kilometers away. That's pretty darn close, right? So this is when they aim missions to arrive, right? So you launch a satellite so that it takes the shortest path possible to get to Mars, and it requires the least change in its velocity to get there. So, oh, this is fun. What's it like on Mars? Right now, according to the Mars Science Laboratory, it's September, at least the equivalent weather to Earth. It's about -75°C, where Curiosity is hanging out. And the pressure right there is about 0.0073 atmospheres. So we're talking about 1% of the Earth's atmospheric pressure. So despite everybody talking about complex atmospheric conditions on Mars, you have to realize that this density is actually very low compared to what we're used to here, even though there's still complex processes occurring there. And there's the comparison to Earth at 30,000 feet that most of you flew at recently. Click. There we go. Composition of the Martian atmosphere, which I'm sure somebody will talk about in more detail later, mostly CO2, a little argon, a little nitrogen, as opposed to us, mostly nitrogen. Right? All right, the different missions that have been to Mars. We have the Mars Reconnaissance Orbiter, Mars Express, the Spirit and Opportunity rovers, at least one of which is now permanently stationary, very sad, got stuck in some loose soil. MAVEN should arrive next month, I believe, just a couple of weeks from now. The Mars Orbiter, I believe it's called, and the Mars Science Laboratory. So each of these carries a suite of instruments on them. They can do different things. I will put up the different instruments here and just highlight the ones that I think we're going to hear the most about over the next few days, or that I have heard the most about recently. And on the Mars Reconnaissance Orbiter, that one is definitely CRISM here. All right, so this is a reflectance infrared spectrometer, so it's just looking at the light that is impacting the surface of the planet or coming off the surface of the planet, takes the spectrum in the near and mid-infrared region. Now, in these regions, this is very sensitive to the physical structure of the minerals on the surface. So it gives you an idea of the hydration state of these minerals and the different types of species which map out the entirety of the planet. Mars Science Laboratory can scoop up some dirt and look at it, but it's limited to where it's at. This thing sweeps huge swaths of the planet at 18-meter resolution, which is pretty impressive. There's also a ground-penetrating radar system on here that I think we'll hear something about later on in the conference. Complementary to CRISM on Mars Express is OMEGA. So where CRISM was 18-meter resolution, OMEGA is 100-meter resolution, and it covers similar ranges. So OMEGA is sweeping out the entire planet at low resolution and identifying places that might be of interest to zoom in on with CRISM, right, to look at rocky outcroppings and differentiation on the surface. Mars Orbiter here, it's called the Mars Orbiter, has a few energetic radiation sources detectors here, as well as a visible camera at relatively impressive resolution of 18 meters again. So it's just taking pictures of the surface and mapping it out. And MAVEN, which will arrive, has neutral gas and ion mass spectrometer, imaging ultraviolet spectrometer, and energetic particle analyses here. This is going to do a lot of work on the Martian atmosphere and the radiation environment there. And Spirit and Opportunity on the planet. As I said, they've been there for quite a while. One of them is now stuck, but they take pictures. They can scrape up rocks. They can measure the magnetic field of Mars. They've been invaluable. And then most recently, of course, is the Mars Science Laboratory, and one of the instruments here is the Rover Environmental Monitoring Station. That's where I got the weather forecast for Mars, and we'll hear something about that from somebody soon. It also has a frickin' laser attached to its head, so it can blow something up 7 meters away and then take the spectrum of that, which is just cool. 7 meters, it's fantastic. All right, moving on to Europa in the last few minutes here. Satellite of Jupiter. It's just a little bit smaller than our moon, not much. And if you were to measure the, well, that didn't translate. If you were to measure the density of Europa, there's a whole bunch of text here, believe it or not, you'd find that it's actually relatively dense compared to what you might originally think. And that density leads people to believe that it actually has a solid or a molten iron core, and then a water-type ocean, right, with a silicate mantle. So there's actually a rocky mantle on top of that core, between the core and the ocean, and then a crust of ice over the top, at least according to the papers I read, which are a few years old. Also, it's because there's no black background here for some reason. That's going to be a problem. Hubble has observed plumes of water spouting off the surface of this planet. Right? Now, this is not just a little bit of water, this is a ton of water. It comes out in amounts of 7,000 kilograms every second of water shooting as high as the Himalaya mountains, I believe, off the surface of this planet. These are giant plumes of water coming off the surface of Europa, which is really cool. Europa's surface is also very smooth, and it's a very young surface, and that's apparently caused by its interaction with Jupiter. So Jupiter here induces a magnetic field, small one in Europa, and it also exerts extreme tidal forces on it. So it's constantly reshaping the surface of Europa to be quite smooth and relatively featureless, with the exception of these things, which the word is there, Linnae. There were a lot of talks on these last year at AbGradCon. I don't know if we're going to see a bunch this year. These are off-color bands, depressions in the surface. They're more than 20 kilometers wide in some cases. That's as wide or wider than the Grand Canyon. All right, these are huge crosses across the surface of Europa. And one of the leading theories is that they're caused by tidal forces from Jupiter, causing ocean currents to come up and split the surface ice, right, and spread it apart into these wide chasms. And so you have differentiation in the age of the ice and also the mineral composition as it's dragging stuff up from the bottom of the ocean caused by this turbulence. And as a cartoon from Wikipedia again, that ocean that I'm talking about, apparently, there is still some debate whether or not it's actually liquid under there or whether or not it's just a warm, moving body of ice, as opposed to the cold, solid surface ice on the top. So something more akin to a glacier here underneath that's just being slowly torqued about by Jupiter's influence. And the tidal forces from Jupiter are 1,000 times that of the tidal forces from Earth on the moon. All right, so it's a huge influx of energy into the system. These are the different satellites, which again didn't make it onto the PDF. So originally imaged by the Pioneer 10 and 11 satellites, 11 and 12 that went by. Voyager 1 and 2 caught some information as it went by. We also have Galileo, which was around Jupiter until 2003, and New Horizons just sent back some high-resolution images on its way to Pluto. And that's it. Enjoy AbGradCon.